Hello everyone and welcome to our channel. Medical authorities and scientists are now in a race against time as they respond to the growing 2026 Ebola outbreak in Central Africa. By May 20th, health agencies had reported more than 600 suspected cases and at least 139 deaths. The World Health Organization, or WHO, has officially declared the outbreak in the Democratic Republic of the Congo and Uganda a public health emergency of international concern, although officials say it still does not meet the definition of a global pandemic. The outbreak is being driven by the rare Bundibugyo strain, not the well-known Zaire Ebola virus, which caused several major outbreaks in the past decade. The Bundibugyo virus has a mortality rate of around 25% to 40%, lower than other types of Ebola, which average around 50% to 60%. However, because the genetic sequences for their outer spike proteins differ by roughly 30%, the approved highly effective Zaire vaccine, Ervebo, does not cross-protect against Bundibugyo. Also, the FDA-approved monoclonal antibodies, Inmazeb and Ebenga, are specific for Zaire Ebola virus and would likely have limited efficacy against Bundibugyo Ebola viruses. Additionally, unlike Zaire Ebola, which has been extensively studied through human clinical trials, Bundibugyo Ebola has very little human treatment data available. Scientists therefore must rely heavily on experimental therapies and preclinical research conducted in animals. To rapidly generate human data during the current emergency, the WHO is fast-tracking a major clinical trial to evaluate two experimental treatments, MBP-134 and remdesivir. Researchers are moving quickly to deploy these interventions under randomized controlled trial protocols in affected regions. One of the most closely watched therapies is MBP-134, a two-antibody cocktail that has shown remarkable results in laboratory and animal studies. A landmark 2019 study published in the journal Cell Host and Microbe demonstrated that this therapy could fully protect ferrets and non-human primates against multiple Ebola species, using a single low-dose treatment. The therapy combines two broadly neutralizing human monoclonal antibodies called ADI-15878AF and ADI-23774AF. These antibodies target different parts of the Ebola virus glycoprotein, allowing them to attack multiple viral forms at the same time. Scientists also engineered the antibodies to enhance their immune system activity through improved Fc effector functions. What makes MBP-134 especially important is its broad-spectrum potential. Rather than targeting only one Ebola species, it appears capable of neutralizing several dangerous strains, including Zaire, Sudan, Bundibugyo, and even related viruses such as Bomali virus. In ferret studies, the therapy achieved complete protection against lethal infection. In non-human primates, a single 25 mg per kilogram dose provided full therapeutic protection across all three major Ebola species tested. Researchers say this could represent a breakthrough because this broad pan-Ebola strategy could eventually replace older strain-specific therapies. That means one MBP-134 therapy may eventually help fight multiple Ebola outbreaks at once. Another drug being urgently evaluated in this trial is remdesivir. Many viewers may recognize the name from the COVID-19 pandemic. Developed by Gilead Sciences, remdesivir was previously tested during the 2018 and 2019 Ebola outbreak caused by the Zaire strain. Later, it was also used globally during the SARS-CoV-2 pandemic, although its effectiveness there was considered modest. Now scientists are revisiting the antiviral because laboratory studies suggest it may actually show stronger activity against Bundibugyo Ebola than against the Zaire strain. Early cell culture experiments indicate the drug has higher in vitro potency against Bundibugyo virus, raising hopes that it could become part of an effective treatment strategy. To organize these emergency studies, researchers are using what is known as the Partners Protocol. This clinical trial framework was originally designed by the University of Oxford for rapid deployment during dangerous filovirus outbreaks. Scientists have now updated the protocol specifically for Bundibugyo Ebola. The trial is currently awaiting final ethical and regulatory approvals from both the Democratic Republic of the Congo and Uganda before patient enrollment officially begins. Health officials say this process is critical because reliable historical data for the Bundibugyo strain remains extremely limited. At the same time, several additional experimental therapies are showing promise in preclinical research. One important example is a monoclonal antibody called Bundibugyo This antibody was originally isolated from survivors of Bundibugyo Ebola infection. In a 2018 study published in the Journal of Infectious Diseases, researchers tested the therapy in rhesus macaques infected with lethal Bundibugyo virus. The results were striking. Treatment with Bundibugyo provided up to 100% protection, even when therapy began as late as 8 days after infection. Another promising pan-Ebola virus experimental therapy is the REBOV-515 and REBOV-442 antibody cocktail. A 2021 paper published in the journal Cell described how these two antibodies, isolated from Ebola survivors, target separate regions of the virus glycoprotein. Together, they produce strong synergistic neutralization against multiple Ebola species, including Zaire, Sudan, and Bundibugyo Ebola viruses. Scientists say the therapy also appears resistant to viral escape mutations, a major concern during rapidly evolving outbreaks. Vaccine studies are also receiving intense attention. A 2023 publication examined whether a recombinant vesicular stomatitis virus-based vaccine could provide therapeutic protection against Bundibugyo Ebola. Researchers administered the vaccine to 6 cynomolgus macaques only 20 to 23 minutes after infection. 5 of the 6 treated animals survived. That represented an 83% survival rate, compared with an expected natural survival rate of only 21% in this animal model. Even more encouraging results emerged from a 2024 study examining trivalent recombinant glycoprotein vaccines that included antigens from Zaire, Sudan, and Bundibugyo Ebola viruses. Researchers tested both single-strain and multi-strain vaccine formulations, often combined with adjuvants and lipid nanoparticle delivery systems. The vaccines generated broad antibody responses and strong T-cell immunity across multiple Ebola species. Scientists found that combining glycoproteins from different viruses significantly expanded immune protection. Researchers say such a pan-Ebola virus vaccine would provide broader and more practical protection than current single-strain vector vaccines, helping close major preparedness gaps for future outbreaks involving Sudan or Bundibugyo Ebola. Still, until targeted therapies and vaccines become available, public health officials say the world must rely on the traditional basics of Ebola control. That includes aggressive contact tracing, rapid identification of infected individuals, safe patient isolation, and strict infection prevention procedures in hospitals and communities. According to the US CDC and the WHO, Ebola patients often die from severe shock, liver and kidney failure, and massive fluid loss caused by relentless vomiting and diarrhea. However, many patients can survive when they receive rapid, supportive medical care. Current treatment guidelines focus heavily on aggressive, supportive therapy. Healthcare workers administer oral and intravenous fluids, replace critical electrolytes, stabilize blood pressure, control pain and fever, reduce vomiting and diarrhea, and quickly treat any secondary infections that strike the patient's weakened immune system. For now, the world is watching closely as scientists push experimental therapies from the laboratory into emergency clinical trials. The outcome of these efforts may not only shape the response to the current Bundibugyo outbreak, but also determine how prepared humanity will be for the next Ebola crisis. That's all for today. If you find this topic interesting and helpful, please give this video a thumbs up, subscribe to the channel, and turn on notifications so you won't miss our future updates. Thanks for watching, and we'll see you next time.